I welcome you for this another sessions on analytical techniques. Today I am going to talk about agarose electrophoresis. What is agarose electrophoresis? We have so many electrophoresis we have seen in previous sessions, like page electrophoresis, rescue page electrophoresis, isoelectrophoresis, all these things. So one more important, a common technique used in molecular biology studies is agarose electrophoresis. So what is this electrophoresis used for? Agarose electrophoresis is a very common technique used to analyze DNA, RNA, protein, mainly based on the size. It is employed mainly in the molecular biology and genetic research, mainly for DNA fragment analysis, PCR product information, DNA sequencing, and more. So before going into this, we should know what is agarose gel, what is it is made up of, what is how it makes the gel. So agarose gel, it is mainly composed of agarose. It is nothing but a natural polysaccharide extracted from seaweed. This agarose is a linear polymer made up of repeating units of agarobiotes, which is a disaccharide. It mainly consists of D-galactose and 3,6-anhydro-L-galactopyranose. So these disaccharides will be repeated to form a linear molecule, that is agarose. The properties of agarose, including its ability to form a gel matrix when heated and then cooled, make it an excellent material for use in gel electrophoresis. So agarose is used widely because of this property. So now we'll see about the steps involved in this agarose electrophoresis. The first thing is preparation of the gel. Normally, the agarose is weighed 0.8% to 2% based on the type of DNA things to be separated. Normally, we use only 0.8%. So based on the need, the concentration of the agarose can be varied. Then the agarose powder, what we weigh, that should be mixed with buffer, suitable buffer. Tris acetate, EDTA buffer, tris borate buffer, EDTA buffer, all these things can be used. After adding the powder into the buffer, we heat this in a microwave or a hot plate. It is mixing it properly so that agarose dissolves completely. After dissolving that, we should cool the mixture, but we should not solidify that. Before solidification, we should pour that content into the tray. And after pouring the content, that is the liquid content of that agarose into the tray, we place the foam and make the gel to be solidified at least for 20 to 30 minutes. Afterwards, then we prepare the tank. After solidification, remove the foam, then keep the gel in the tank. You should add the buffer so that it covers 1 to 2 centimeter above the gel. Normally, the buffer used for agarose gel electrophoresis is tris acetate EDTA buffer, that is TAE buffer, or tris borate EDTA buffer. These buffers mainly responsible for giving the ionic environment for the electrophoresis process. Otherwise, the separation will not be neat. The third step is loading the samples. After removing the foam from the gel, we have the wells created. To these wells, we mix the DNA samples with a loading dye because that makes the sample denser so that it can be visualized. The dye can be used to visualize the movement of the sample. So load the samples into the wells using the micropipette. Then you may want to include a well for the DNA size markers as well, because we should know what is the size of the DNA what we are going to separate. For that, we need a standard, so a marker DNA size markers also run in a well. Then after that, we turn to the power supply so that electrodes are connected properly, positive to positive, negative to negative, then we switch on the current. You should set the desired voltage and the current time. Then based on the size of the DNA fragments and the percentage of the gel, the separation occurs. So the molecules will move towards the positive electrode. After the electrophoresis is complete, after the visualization of the dye to the stage where we need, then we switch off the power supply, remove the gel from the tank. Then after suitable protection of your hand, you handle the gel by staining with ethylene bromide or some other dye which you are using now like cyber green. Then after that, staining the gel, we should visualize using gel top or in UV chamber. Normally nowadays we don't go for sodium bromide, instead of that we are using cyber green because they can be used even for RNA and proteins can be done. So when we cyber green if use, what happens when we UV light is fast or when you keep it in the UV light, the gel, they emit fluorescence. That can be documented and that can be image software. We can take the pictures and we can store it and we can analyze. So afterwards we can de-stain the gel by rinsing it with water to remove the dye also. So this is the setup. You see this, we can see the gel is kept in a tank where the both sides we have tank for the pouring the buffer. And this can be supplied to the power supply. And we have a well where the sample is loaded. When we put the current on, give the supply the current, the gel will be moved and the band will be formed after straining. So the UV transilluminator and otherwise like gel top, we can capture the DNA bands, the imaging using imaging software. Then we can find the distance of the movement of the bands. From that we can estimate the size of the fragments. Very important thing, we should handle the ethylene bromide and other chemicals using the safety protocols to be followed in the lab. So we should dispose these gels in ethylene bromide solution according to the chemical based disposal guidelines. So this is how. So first we take the agarose and mix it in the buffer, then we melt it, then we pour it in the tank tray using the foam. After solidification, remove the foam, load the sample and keep it in the tray, pour the buffer, switch the power on for the electric current, run the gel, strain it and visualize it, then we can analyze it. What are the applications of this Agarose electrophoresis? So mainly we can analyze the DNA fragment because it is used to separate visualize DNA fragments, mainly in PCR, intersection and digest genomic DNA. So all these things we are using this Agarose electrophoresis. And it is also employed to separate and analyze RNA molecules like mRNA, rRNA, tRNA, other small RNA molecules. It's mainly used for studying the gene expression studies, processing and modifications of RNA. So it is also used to study the size-based purification of DNA and RNA fragments. So it is also used to confirm the success of PCR amplification by visualizing the presence and size of the amplified DNA fragments. So even the product formation of PCR can be visualized or can be confirmed by Agarose electrophoresis. Then DNA sequencing. So these are mainly used in initial stages for sequencing, mainly to separate and purify the products of chain termination reactions. After this preliminary work, they can be moved to the capillary electrophoresis system for high throughput screening. But for sequencing, initial work is done in Agarose electrophoresis. Likewise, it is also used to analyze plasmid DNA, like the size of the plasmid and the confirmation of cloning, all these things. And it is also used in genotyping, that is by separating DNA fragments that contain genetic markers associated with some diseases or traits. Then this technique is combined, this technique combines electrophoresis with hybridization, detect specific DNA sequence in the sample like Sjctnc-Dr. The southern blotting, the Agarose is linked with hybridization technique to detect the sequences in the sample. That way we can map the gene DNA fingerprinting, also studying the copy number of the genes. Like microbial fingerprinting, that is, used to generate DNA fingerprints of microbial identification and epidemiological studies. And also for protein DNA interactions, you can use this. And it can also use to immobilize and separate biomolecules, mainly used in affinity chromatography, including purification. Likewise, this can be applied in Northern blotting also to analyze the specific RNA molecules. And it is, while agrogeles are more commonly associated with nucleic acid, sometimes they can also be used for protein analysis, particularly for the separation of native protein complexes based on their size. Thank you. Thank you for listening to this agrogeles.